This is an instructional video on basic use of the Leica SP5 Confocal microscope. And what we're going to go through in this video is a simple one-channel imaging by confocality using a single laser line. Additional functionality of this microscope will be explained in small video modules that will appear in the future in addition to this module. So for today we're just going to cover the startup and the shutdown of this microscope and how to set up for simple confocal imaging and some tips of how to get effective confocal imaging using this device. Right, so let's start with the startup. The device is configured from a level of hardware and a level of software to essentially work from left to right. So to start up the microscope, we're going to turn on the mercury lamp. Then we're going to go over to the panel beside the scope and turn on the PC microscope, the scanner power, the laser power, and we're going to turn on the key lock interface for the laser, which is a safety requirement whenever lasers are involved in equipment. And then we're finally going to turn on the computer that runs the microscope. And so we're going to have a standard Windows startup and login. So once the computer started, we go with our password login. Which will bring us to a standard Windows startup. And in the middle of the screen, we're going to see an icon that's going to launch the confocal microscope. And here's our icon, the LASAF. We'll double click on that icon. And we're going to start the launch procedure with the software. So what's going to happen now is an initialization phase, and that means that the computer is going to start communicating with all the components of the microscope, as most of the microscope is under computer control. And you're going to get a scroll list appearing here of various checks that are occurring on its way to launching the program. It's going to start with initiating the hardware server, and then it's going to say it's going to check the configuration of the machine, and there's going to be an OK or cancel step. We're going to press OK. Now it's going to load the hardware tree, and this is going to start with the communication on all the devices, and you're going to hear various switches getting flicked on and off as the computer interfaces with the microscope. At this stage in the startup, you're going to get a query asking you if you want to initialize the DM16000 stage. And the correct response is no, we're not going to initialize the stage. And then that will continue with the rest of the startup procedure. And the final line that you should see is success, indicating that everything's been properly initiated, and then the software will boot up. And so we're reading here, initializing user interface, and that interface will appear in a few seconds. Upper left-hand corner. So once the software has initialized, the software control works very similar to the hardware control in a left-to-right pattern. So we're going to start with the configuration up here in the left-hand corner. And click on that tab, and click on the laser tab, and turn on the lasers that we're going to be using for this session. So just for the sake of convenience, I'm going to turn on the argon laser, the 561 solid-state laser, and the 633 line helium neon laser. And as soon as you click on the argon laser, you should hear fans turning on, indicating that the laser is started up. And we also have to set the power level for that laser as it starts up, and I recommend approximately 1/3 power. And as soon as we do that, you can see a progress bar going up to that 30%, and this is the warm-up time required for the laser. In the next tile, there's the objective. In this case, we've had this set at the 63x glycerol immersion objective, and that's pretty much for the configuration section, which means we're now going to switch over to the right to the acquire section. All right. So prior to acquisition, I'm going to just set up our slide sample for imaging on the scope. So this device is the remote control that controls the microscope stage. It has buttons for XY precise focus, XY precise movement, XY fast movement, a Z movement with a very fine control and a Z movement with a coarse control. We tip back the head of the scope, and what you can see is with this top and bottom knob, we can translate the stage up and down or left and right. And the smaller knob on the top is what controls the focus upwards and downwards. So for this scope, we currently have two objectives fitted, a 20x air immersion and a 63x glycerol immersion. And this means that all we need for imaging is one drop of glycerol solution that goes right onto the surface of the objective. The slides are mounted inverted so that the cover slip is facing down, and the holder can hold many types of slides or dishes. And we're now going to align the X and Y approximately to the center of the slide, and then with the top knob I'm going to turn it clockwise to bring up the objective until we can just see the contact between the slide and our drop of glycerol. And that's going to be the approximate correct focal plane. I'm going to tilt the head back, and now we're going to do some wide field imaging by fluorescence to set our focal plane. At the front of the scope, there's a display panel here that tells you a lot of information about the microscope. Basically, it's telling us here that we're on our 63x objective, and we have 100% of the signal either going between camera or eye. I'm going to press this button to switch it to the eyepiece, and then we have a choice of two excitation wavelengths for green fluorophores or for red fluorophores. And in this case, I'm going to choose green fluorophore, and you should hear a click as that sets of dichroic filters are put into place, and then the button in the center will control the shutter, which will give us our excitation light. In addition, there's a sliding scale on the side with two buttons here with the label INT for intensity, and that allows us to either increase or decrease the fluorescence intensity as we're scanning by eye. And you really only want to decrease the intensity if you're worried about a sample that could be potentially bleaching while you're trying to find the cells of interest. So now that I have my excitation light in place, I'm going to look through the objectives and come into our correct focal plane. And we're going to look around for cells of interest that are in correct focus. And once I have the correct focus, I'm going to switch it over to Z Fine button, which is now going to give me very fine focus control, get a very sharp image, and we're done. So then I'm going to close the shutter, which is going to protect the sample from any photo bleaching. So once the object has been focused on, and we know we have which is either FITC, we pretty much take over control of the FITC through the software. And what will happen now is a profile setting will get put into place where a laser line is picked, and we have an emission profile for our fluorophores of interest. In this case, it's fluorescein isothiocyanate, very similar to Alexa 488. And what we're getting here is a visual representation of what's going on at the microscope. So we have our laser power here at 48 nanometers of light. The argon laser has multiple lines, 458, 476, 488, 496, and 514. The solid state lasers are 561 and 633 laser wavelengths. So 561 is compatible with most of the reds, such as Texas Reds or CY3, and 633 length is compatible with far red dyes, such as DRAC5 or CY5. These other lines have been more or less tuned for fluorescent proteins. 476 or 548 may be most appropriate for cyan fluorescent proteins. If you just have GFP, 48 is the correct line for that. And for yellow fluorescent proteins, we have a 496 line. And what you'll see is the lasers are controlled for power via these sliders. But the important thing to consider is that not all of the laser lines have the exact same power output. So just because the lines can all be set to approximately 20% power doesn't mean you're getting the same amount of light through the system. To start with, I like to set the power at the 488 at approximately 20% or less. And this is in fact one of the more powerful lines that are on the microscope. So the laser is going to travel down a fiber optic cable and come to a crystal that's known as an AOBS, or an acousto-optical beam splitter. So unlike a conventional microscope that has a dichroic mirror that separates the emission wavelength of light from the excitation wavelength of light, this microscope uses an AOBS. And what that means is, by passing the light through the crystal, controlled by a certain frequency of radio waves, you can actually set which wavelengths of light are allowed to go through the crystal. It's far less restrictive than a dichroic filter. So the line is going to go through the AOBS, go up to the specimen, it's going to excite your fluorophore, and then your emitted light is going to come from the specimen across the AOBS again. So now you're only going to see the emitted light and not the excitation light. And then now, again, unlike most microscopes, instead of passing through an emission filter to a photon multiplier tube, you're going to pass into a graded set of PMT filters that are adjustable. And what that means is, as you can tell here from these sliders, is that we can manually adjust how wide or how tight the emitted light frequency is being captured, and we can judge all this relative to the excitation wavelength, which is shown by this laser line here. What you're seeing displayed in this graph here is the typical emission profile of fluorophores. So we're actually looking at an Leica 488 fluorophore, so I'm going to choose that one from the list. So that is giving us an idea of what our fluorophore emission profile is like. And we're going to set our gating to be approximately right onto the peak of that fluorophore. And once you've done that, you also have options as to how you want the color to appear on the screen. So what you have to remember is, what's going on here is this light is being gated, and it's being gated by frequency and then being recorded by an active photon multiplier tube. And that's what active means here when this checkbox is being highlighted. But what we're really seeing is just an intensity of light. So in reality, the light that's coming out doesn't have color. It's not being seen by color, it's just being seen by its intensity in this part of the wavelengths. So the light is essentially a grayscale. So you can assign any color you want from this list of lookup tables for the fluorophore of interest. Typically, if you're using green, you tend to choose a green lookup table. If you're looking at a red fluorophore, these days for modern publishing, we tend to look at a magenta lookup table. But for our cases here, we're going to just deal with one fluorophore. In another module, we're going to talk about multi color imaging. Okay, so once we've chosen our fluorophore and our pathway, the next step is to now look at our acquisition mode. And this is back on the left-hand side again. And what you can see is we have two bars here representing the pixel array, which is set at the default 512 by 512, and the scan speed, which is set at 400 Hertz. Now the pixel array can be increased all the way up to 8,000 by 8,000 pixels, and the speed of scanning can vary from a very slow 10 Hertz all the way up to 1400 Hertz. Typically, for most panels, sub-panels required for publication in journals, 512 by 512 is more than sufficient. If you're interested in some particularly high resolution imaging, you may want to switch this up to 1024 by 1024. If you're going for journal cover art, you may consider 2000 by 2000 pixels. The speed is important to consider. You want enough speed so that you aren't taking a long time during the acquisition process and encouraging bleaching, but you have to remember that as you increase your scanning speed, you're going to also increase the amount of noise that hits those photon multiplier tubes. So typically speaking, we start at around 400 Hertz, and when it comes time to tune the image, we'll either increase or decrease that. If you're interested in live cell imaging, typically you want to scan at 400 Hertz or better. So we've chosen our scan speed, and we have our light path set up for our green fluorophore. And so for the next step is now to take a scan of the image. And so there's 3 buttons that appear down in the bottom of the software. One is the live button, the capture image button, and then finally the start button. The live button literally starts just scanning live and will continuously scan as long as this button is depressed. So when I press live, you'll see a red light appear there, and what we're seeing is the continual scanning of our sample. And any manipulations that we now do in terms of focus or position using the remote control will be recorded in real time as we're scanning. Of course, we don't want to do too much of that, because every scan that we put onto this sample is exciting that fluorophore and is going to start triggering the photobleaching process and degradation of the signal. Right? So from our first live scan images, we get a nice representation of what we see through the eye field. But typically at single cell imaging, we want to focus in even tighter than that. So we then switch to the next section of this pull-out menu, and we're interested in zooming in on a sub-region of this field. So what happens, if you look carefully over at this menu, when I click the checkbox for zoom in, immediately what happens is a region of interest box appears. And if I take the region of interest box and draw it around any object on the field, every subsequent scan will just be to that region of interest. And I'll show that by pressing live briefly. And what we've now done is focused our scanning just on this region. So you have to consider now that the smaller you make your region of interest, for a given laser power, the more laser energy is going to be focused on your area. So as you get into smaller and smaller regions of interest, you are going to increase the level of photobleaching occurring in your sample, right? So we now have to look at the settings that are being captured for our imaging. In this panel, if we click on it, on our display underneath the display monitor for the image, we're going to see a series of readouts and knobs. And so from left to right here, we have a setting of the gain. And that's how much amplification is occurring on that photon multiplier tube. This has a maximum of about 1250. And when you hit that maximum, you're going to hear that alarm. So you really can't exceed that. Now what you have to remember about the gain is that the more amplification you have of your signal, the more contribution noise will have to your signal. So just increase, it's a good to start by setting the gain pretty much near maximum, but we're probably going to want to back down on that for high quality imaging. The next 2 buttons I don't recommend most people ever use. This is the offset button and the scan field rotate button, and this is pretty much not, you really don't need to adjust this from the default settings. The next adjuster is important, and this is for the pinhole size. Confocal imaging achieves its confocality by cutting out the out-of-focus light. It's essentially the same as a wide-field inverted microscope, except that pinholes are now in place above and below the object, and the out-of-focus light is excluded from hitting the PMTs. But using this dial, we can adjust how big or how small that pinhole is. The smaller the pinhole, the less confocal volume you're seeing, the less light that you're getting through. The larger you make the pinhole, the more light that you get through, but the less confocality you have because you have a larger volume of information coming towards you. So I'll show you by example. Currently we have the pinhole set to approximately 100 micrometers. If I open up that pinhole to 200 micrometers, having all the other equivalent settings, and I do a live scan, you can see that it's dramatically brighter. And it's dramatically brighter because we're now allowing more light into the system. Now you can also see that in addition to this brightness, we have a large amount of noise in the background that appears as this snowy background on what should be an entirely black background. So now we can start tuning this image. So clearly it looks like the image is too bright, but there's one way to tell that for sure. Up in the top left-hand corner, there's an option of different palettes in which you can visualize your signal. So if we click on this lookup table, this quick LUT, we can switch between intensity on a scale, black and white imaging, or the color that we have assigned. And it's really good to start your imaging by looking at the intensity scale. And what this is a lookup table that goes from white to red in terms of the linear range of the photomultiplier tube. If your pixel intensity is exceeding that linear range, you then see the appearance of blue pixels. So this is telling us that the bright image that we have on screen is in fact exceeding the dynamic range of the photon multiplier tube. And what we really want to do for the sake of being quantitative and for the sake of accuracy is get our signal below that upper dynamic range. So what we're going to do is therefore adjust our settings until this comes down. And so I'm going to press live scan, and I'm now going to back off on the gain until we no longer see blue pixels, which is approximately there. And I press stop. So it looks like the proper gain setting for this channel is about 820.4. Now, another potential way to do this is by adjusting the laser output. So if I decided that my signal to noise was fine for the gain setting that I had, I could decide to turn down the laser. That's usually the preferred route to do this because as you turn down the laser, you minimize photo bleaching. So you can see when I reduce the gain down to 820, I now achieved an absolutely black background with absolutely low noise, which says that this is the optimal setting. We are now in the dynamic range, so we can now choose to look at this image either as a grayscale intensity image or as a color image. Now you have to remember also what's being displayed here is the full linear range of your pixels. In reality, you can adjust this post-acquisition on software such as ImageJ. So you in fact have a lot more image intensity here than you think. And I'll demonstrate this for you. So we're going to leave the gain settings where they are. We're going to turn down the laser power down from 20 to about 7%, and I'm going to do one quick scan. Now, to do a quick scan, I can do live and then quickly turn it off. Or if I want to do a single scan, I can press capture image. Okay, so what you can see is now I've captured the image, I've turned down the laser intensity, and this has given me a relatively dim image. And this is actually probably the intensity you should be acquiring at, because if you go to these sliders that appear on the side here, what they're doing is giving you a readout of where the range is of your data. And as you can see, by adjusting the slider from either end, there's actually a tremendous amount of pixel information in this image. And as you can see, as I crank it up considerably, we now start to see the appearance of some noise, which can be reduced by thresholding in the opposite direction. So by looking at this, we can see that in fact we do have a lot of light information here, even though it's not displayed this way, and we care to look at this across the whole image. And I'm going to just set this a little bit brighter, so I'm going to set our intensity back up again, and once again I'm going to press capture image. So this is at about 15% laser power. Now, every time I press capture image, what happens over here up in the corner under the experiments tab is that file is being generated. When I'm doing live scanning, no file is being generated. That data is not being saved. When I press capture image, a file is being written, but it's not being named. So what you have to do is either at the end of the session or preferably during the session is to right click on that image and save it under a different name. You also, at the end of the experiment, have the option of saving it as a variety of different file formats. The standard format that comes off of the microscope is LIF, which is a Leica like TIFF format, so it's completely lossless. It contains all the raw data information, including all the microscope settings. And this is the setting that I recommend that you save your raw data in. From that, you can generate TIFF files, and in other software, you can go from TIFF to almost any other file format. Once you've started the experiment, you can occasionally hit the button down here that says Save All to ensure that you've saved all of your experiments. If you try and shut down the software prematurely, it will prompt you and ask you if you want to save. But it's a good idea as you go along, because with any high-performance piece of scientific equipment, you know, there are crashes, there are bugs that can occur. You want to make sure that you're not losing any data and wasting time by having to do it all over again. So back to the acquisition plane, we can see that I've captured this image. Now, for the sake of this video, I'm going to turn up the signal so it's a little bit more intense and turn up the gain. And I'm going to press capture image. So we're going to get a nice bright signal. Now, this may or may not come across in the video, but there are some artifacts that are appearing in the imaging as a result of relatively fast scanning at 400 Hertz. So what I'm going to do is slow down the scan speed to 100 Hertz. And you can immediately see this when you capture the image button. It's going to take 4 times longer for that scan to now occur. But what you'll also notice is that you're going to get a higher quality image. Your signal to noise ratio is going to be much higher, and you're going to lose any artifacts of scanning of the laser. And generally speaking, you're going to get a much higher quality image. However, as that laser is scanning slower over the region of interest, you're also going to increase photobleaching. For most modern fluorophores, such as Alexa-based dyes, these dyes tend to be extremely good for photobleaching and photoresistance to bleaching. But for live cell imaging, this is more of a concern when we're using fluorescent proteins. Another way to get a higher quality image is to average the image. And so if we go back over to this panel here, you'll see options for a line average or a frame average. Basically what averaging is for each scan that occurs over the image, it's going to overlay the scans on top of each other, and it's going to give you, therefore, destruction of noise and increased or construction of signal to give you a better signal to noise ratio. So for this image, I'm going to set the frame averaging to 4 frames. And what I'm now going to do is either press capture image or press start. So what's the difference between these two buttons? Capture image just captures an image, a scan, and writes a file on whatever current settings are on the microscope. Start is what occurs if you have an experiment that includes many different settings in the microscope at different stages, and we'll cover that in subsequent modules. And this is going to include things like multicolor imaging, stacking in Z-plane, or fluorescence recovery after photobleaching, or Forster resonance energy transfer. So I'm going to press the capture image button, and now what's going to happen is you're going to capture 1, 2, 3, and then a 4th scan. And what you can see when you're at the scope, it might not be so obvious in the video, is with every scan and with every overlay, we're just improving our signal to noise by that much until we get approximately our highest quality imaging. We've got a very robust signal. We have absolutely no visible noise. If I go over to our lookup table, we're just starting to have some pixels outside of the linear range. So I'll probably want to turn down my gain a little bit. And I'm going to capture this image again. Okay, so now our pixels are back within the linear range, and we're doing the 4-frame averaging of interpolation to get our highest quality image. Typically what's going to happen in this type of imaging, if you're not using immunofluorescence, you're more than likely going to be faced with the problem of how to increase the amount of signal that you want to see. And this can be done by a variety of methods. One method is to simply turn up the laser power, and I'll demonstrate the caveat of that in a second. The other option is to increase the gain as much as possible and increase the pinhole size as much as possible. We didn't talk about the other two adjusters, but there's an adjuster right beside the pinhole adjuster. So I'm going to turn the frame averaging off to demonstrate this, and I'm going to press live. And what this allows us to do is zoom in manually to the center of the image, or zoom out, whatever we choose. And the adjuster next to that is the Z position adjuster. So once again, we really don't need to touch the microscope at this point. I can press get a live scan, and now we're at 400 Hertz, and I can adjust our focal plane. As you can see, I was slightly out on the focal plane, because now that I'm in the correct focal plane, I've exceeded the dynamic range again, which means I need to turn down either the laser power or the gain on the PMT. So I just turn down the gain now until I see no more blue pixels, and I'll press stop. And now we're in a good position to say capture image, and we can get a very high quality image even when we're scanning as quickly as 400 Hertz. So in addition, if this still wasn't bright enough, I could once again turn up the pinhole size, and we'll turn it up now to 300 micrometers, and in fact we're going to get a much brighter image. But again, you're going to have to check to make sure you're still in the linear range of your pixels, and for the most part we are. I see a few blue pixels there, but otherwise we're fine. And this is going to give us a nice single shot of our cell of interest. So what I'm going to demonstrate next is exactly how much photobleaching can occur in this sample. So if we go back to our controller here for our zoom factor, which is set at 3.58, I can push that slider until it says 1. So we're no longer zoomed out, and I'm going to do a single capture image. And if you look carefully in this capture image, on these cells that surrounded our region of interest that we were repeatedly scanning, they're in fact outside of the linear range, because those fluorophores have never been excited before. But in our region of interest, you can see clear examples of photobleaching of our sample. So photobleaching can occur, and it can occur with a very good fluorophore at just 26% laser power. So there is more than enough laser power on all of these lines to bleach your sample. Like with any good experiment, what you're going to decide on ahead of time before an imaging experiment are which are the best fluorophores to choose for your application, and you may be limited by your ability to choose fluorophores. What some of you will notice is that if you're doing typical multicolor imaging and like to put in dyes such as Hushed or DAPI to look at DNA within the nucleus, we don't have ultraviolet laser lines on this device. But what we do have is a far red laser line. And so there are dyes available that one can use that will allow you to stain the nucleus by using the far red laser line instead of having to use more conventional dyes like Hushed or DAPI. But that'll be covered in another module. Okay, so when working with your set of fluorophores in your experiments, you might find the need that to make your own profile for your own either unique fluorophore or the particular settings that you have for your experiment. And I'm just going to quickly demonstrate how to do this. So I'm going to choose any one of these fluorophores. Currently we have the FITC profile set, and we're now going to modify that profile. So what I'm going to do is I'm going to actually set this up for a red channel. So I'm going to turn off the 488 laser, and I'm going to turn on the 561 line of laser. I'm going to change our, so this is going to indicate where our laser line is here, and it's going to show a yellow color, which is what 561 nanometers is. I'm going to change our PMT, our active PMT, from one at this level of the range to next door PMT3 at this level of the range, and I'm going to just spread out the gating, and I'm going to load the fluorophore of interest, which in this sample is a Leica SP5 right? And what that's going to do is give us a profile of the Leica SP5 dye. And this gives us an approximate measure of how wide we should be able to select the gating to see the fluorophore. And I'm going to give it a color. And the color is any one of these lookup table colors, whatever you want. For modern journals, they're now insisting that instead of using green, red, and a merged color of yellow, that the appropriate color to use is in fact magenta. And that's for a variety of reasons. Magenta prints better in print journals, and green and magenta are two colors that can be seen by people who have typical color blindness. So we'll say OK to that. And so now what happens is we have a setting which has a 561 exciter across the AOBS to the objective. The emitted red light is now going to come to the spectral grate, and we're going to choose to look at the light between these two wavelengths away from the excited line and approximately near the peak of this fluorophore, and we're going to assign a color of magenta to it. So we've set now the laser line. It's going to go up to across the acousto-optical beam splitter to the specimen. The red light is going to come across to the spectral grate, where we have set our limits to the light that we're going to see, and we have it going to PMT3 with this magenta color of light, a color that's going to be given onto the image. And over here is where the image is going to be cast, and we're going to set our gain on our PMTs. The default on new channels is a gain of 0. So if you're not seeing any light, first thing you need to check is the gain. So what I'm going to do is turn it up to its maximum, which on this photon multiplier tube is 1000 volts. And then I'm going to press live. And this is what I thought I should be seeing, which is a nice image of mitochondria. But as again with the other experiments, we're going to use our dynamic range indicator in our lookup table, and in fact we're very much outside the linear range. So I'm going to fix that by pressing live, and as I'm scanning, I'm going to turn down the gain until those blue pixels disappear. And I'm also going to adjust the zoom a little bit so I can get in on one cell, and I'm going to adjust the Z range to know that I'm at the exact focal plane I need to be, and it turns out I was right from the beginning. And this is approximately correct. So now I can decide to do the rest of my imaging either in grayscale, which is what actually I prefer, or the magenta scale, and zoom in on a single cell and the mitochondria within that cell. I'll press capture image, and now I'm only going to get my region of interest, and I've got a nice delineation of these mitochondria. I've got a good signal to noise ratio. I can see no noise in the image, which is actually telling me that maybe I can tolerate using slightly less excited light, or actually I might want to increase my confocality by closing up my pinhole to about 200. Let's capture that image. So now in fact we're getting a much crisper image because my pinhole is smaller, so we're getting even less out of focus light. And if I really want to improve the quality of this, I'll change my scan speed down to, let's say, 100 Hertz. And if that's insufficient, I'll increase my frame averaging, and let's say for 3 frames of averaging. Now at any time if you set up any of these scans and they take a certain amount of time and you can see that something's gone wrong, either the focal plane is wrong or this is not what you wanted to do, there's always the option of pressing the stop button, and that's going to stop the experiment, right? So every time I press capture image, over here in the experiments tab you can see all these files have been written that show me my images, and if I click through them they'll appear on the monitor, so I can get a quick library of everything we've done so far. Right? Okay, so that's going to end, and so once we've done, once we have our settings for our new fluorophore of interest, we can now press save on this, and we can change the name of it to our particular name that we need to, and save it to the into the settings, and then it'll appear in this long list of settings in the future. Similarly, with your experiments, when you press save, it's going to prompt you to where you want to save. Right? Completed our experiments, and we have our list of images, which you can either name during the experiment or at the end of the experiment. When we press save all, we're going to get prompted to the location of where we're going to save these files. So these files should be saved on the hard drive, which is labeled as the C drive, and under a folder MBF users, and there should be a folder appearing for your name. If you don't have one, create a folder, and inside of that folder you want to save the experiment. And typically it's good to pick a standard format for your experiments and save always in that same format. And the format I prefer is to use the date. So I'll indicate the experiment as 2014 0203, and then I'll put some note onto this where I'll just write in like test or training or something like that, and we'll save the file. So we are not responsible for the data that's saved onto this microscope. It can be used as a temporary storage space for your data, but I strongly recommend that you show up to your session with either a USB key or preferably a one-time write DVD-ROM that you can use in our DVD burner and save the data as a one-time write data. And this will ensure that if you're using the LIF image format and a one-time write to the DVD, that you've got very good data integrity. That data can never be modified in the future. And I think this should be actually the policy of most labs that are doing any cell biology on this device. And we expect that people are going to save the data on their own independent device. Now that could be a USB key, it could be a USB hard drive, or it could be some type of disk media that you can burn and that you're going to take it away from them. We will allow temporary storage on the device, but I will be clearing the device occasionally once the hard drive gets full, because we're not going to be storing device or take responsibility for device storing data or take responsibility for data that's stored on. Once we've finished our confocal imaging session, it's now time to properly shut down this device. So over in experiments, you should at some point during the experimental process press the Save All tab, and shutting down is very similar to just shutting down a computer. So the first thing we want to do is manually turn off the lasers. If we close the window, we'll be prompted to ask if we want to save our experiment, and you can choose either yes or no. And then the Leica software is going to shut down. And once that's shut down, we're then going to shut down Windows. While the Windows is shutting down, we then can go in the opposite direction of startup. So when we started up the computer, we started with the mercury lamp, we went to the PC microscope, the scanner power, the laser power, and then the key lock interface for safety for the laser. Now that the computer is off, we can turn off the computer, we can turn off the key lock interface, the scanner power, and the PC microscope power. Now you note I did not turn off the laser power immediately. This is because, as you can hear, there's a fan running in the background. That fan is actually cooling those lasers. We don't exactly want to turn off the power to the entire laser unit before the lasers have cooled off. So we'll turn off the lasers, but then we'll also keep the power onto that fan unit for approximately 5 minutes or so. If you know that another user is coming into the facility, just leave that on. If you're worried, by default, just leave it on. The last thing we want to do, and very important not to forget, is to turn off the mercury lamp. Because as you will notice during the session, these mercury bulbs get extremely hot, and they have a finite lifespan and will burn out. Okay, so once we've shut down the hardware and the software, the very last step is that we want to remove our sample, slide. We'll give the surface of the objective a quick wipe to remove any of the glycerol solution, and we'll give it a quick spray with a cleaner, and this will ensure that no glycerol dries down onto the surface of the objective, and for the next user they're going to have a perfectly clean objective to work with. We'll then tilt back the head of the scope and replace the cover. And that's pretty much it for how to shut down the Sp5 confocal microscope.